Howdy, folks. Thanks for checking in to Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. And look around. I mean, whoa. This is lush. This is green. Yeah, in this little notes class, we are going to be talking about photosynthesis, how we can get life from thin air. To run through some of the basic terminology, let's take a look at the relationship between life and energy. We can take most organisms and put them into one of two categories. They'll either be heterotrophs, like us animals, which have to consume other organisms for our organic molecules, which we then use as a source of energy through respiration, or autotrophs. Those are the plants, but an awful lot of bacteria and protists and all kinds of other stuff as well. But they're still going to capture energy. The difference here is they're capturing light energy and then using that energy to take carbon dioxide and build, which is inorganic, and build organic molecules out of it. By making these organic molecules, they're actually making energy that's usable by not only themselves, but they're also producing energy, and that's why we call these producers energy for the consumers, for us. So this process of photosynthesis is directly fueling respiration. We have to remember that even though autotrophs go through photosynthesis, all living things go through respiration. And some of that sugar that is produced through photosynthesis is going to be used by the plant for energy needs through respiration. It's important that we're able to explain the connection between heterotrophs and autotrophs a little bit more specifically, a little bit more metabolically. Heterotrophs are only going through cellular respiration. So they're taking that glucose that they've gotten through eating other organisms for their organic molecules and converting that into carbon dioxide and water. This is oxidative. And is exergonic. That's what we're trying to get. We're trying to get the ATP out of it, so we're releasing the energy. Now, autotrophs, though they are going through cellular respiration, they are also going through photosynthesis. Now, photosynthesis, when we look at the overall chemical reaction, is the exact reverse of cellular respiration, aerobic cellular respiration. The only difference is the form of energy. And that's important. So these autotrophs take the light energy and use that as the source of energy for the endergonic reaction of fixing the carbon dioxide, which is inorganic, into glucose, which is organic. And this is, like I said, the kind of, it's not magic, but it's the chemistry that autotrophs are able to do in converting light energy into chemical potential. Let's say you wake up tomorrow morning and you find yourself to be a plant. Well, you're going to need to be able to do a few things that you can't really do right now. One of which is have a way to collect light energy so that you can convert that into a chemical energy form, ATP, that's usable by the cell. But ATP is a terrible storage molecule. It's too unstable. So you need to be able to take that ATP or the energy in ATP and use it to make other storage molecules that are a little more stable, like glucose, and we can move that around. But even so, you're still going to need to be able to get all of those building block atoms, the chans, the carbon, hydrogen, oxygen, nitrogen, and the rest of them from the environment. You can get the carbon dioxide from the air, and hopefully water will be available, but we got to get the other nitrogen and potassium and phosphorus type of nutrients. Because you're also going to need to have all of the cellular machinery needed to make all of the other organic molecules of life, because remember, you're an autotroph. So you need to be able to make all your carbohydrates, all of your proteins, all of your lipids, all of your nucleic acids, the whole 9 yards. That means that structurally, you're going to have some parts to do these things. The sunlight, we're going to get largely through the leaves. Those are the solar collectors. The carbon dioxide is largely going to come through structures within the leaves called stomates, which we'll talk about here in a minute. Water is going to come through the roots, and again, that's going to happen through osmosis and all of those osmotic potential things that we've been talking about. Nutrients as well, those are going to be taken in through the roots. If we're going to take time to talk about photosynthesis, we have to take some time to talk about the structure of the leaf, since that's really where photosynthesis is taking place. So when we look at the cross section of the leaf here, we see that the middle of the leaf, called the mesophyll, literally middle of the leaf, has two layers. The top layer up here is called the palisade layer. It's densely packed. It's where a lot of photosynthesis is taking place. Down below this is the spongy mesophyll. Spongy mesophyll is called spongy because there's all of these air spaces. And what that's going to allow is it's going to allow water to evaporate out of the cells into the airspace. And then because of these stomates, it's going to allow that water vapor to make its way into the outside world. And that's going to complete transpiration. That's what's really driving this whole process of pulling water up from the roots up to the leaves. So a stomate has two cells. They're called guard cells. As the guard cells take on water, if there's plenty of water around, that causes them to elongate and open up, and that allows the water vapor to escape out of the leaf. But that's also going to allow those cells to dry out. As the guard cells have less and less water in, they shorten, and that closes the stomate and allows water to accumulate as it comes up the vascular tissue here. This also is going to allow for gas exchange. So as they open up, it's going to allow carbon dioxide, which is needed by the plant, into the cells so that they have the raw material for photosynthesis. Those are the leaves. If we really want to get into where photosynthesis is actually taking place, we're going to have to get into the individual cells themselves and get right down into the chloroplasts. It's within the chloroplast that we're going to find those photosynthetic pigments, chlorophyll and others. It's those pigments that are going to allow the chloroplast to absorb the sun's light and use that energy to fix carbon dioxide into sugars. In order for us to have a good handle on how chloroplasts are able to carry out photosynthesis, we should really have an understanding of the structure of the chloroplast itself. So it is a double membrane structure. That means it has an outer membrane and an inner membrane. In this case, the inner membrane is broken up into smaller compartments called thylakoids. And those thylakoid sacs are kind of stacked up into structures called grana. Between the grana and the outer membrane is a fluid-filled space referred to as the stroma. Now, the thylakoid membrane is really where the action is happening in that chloroplast itself. Stuck in that membrane, we're going to find the photocenters. This is where we are going to have the chlorophyll molecules as well as some of the other pigments that we find. We're also going to find some of the very same types of proteins, transport proteins that we find in the electron transport chain. And we're also going to find some ATP synthase. This should all sound familiar. So when we take a look at this, here's a very, very large thylakoid sac individual within a chloroplast. So what we're going to do is we're going to build up a hydrogen gradient because we've got that ATP synthase in there, and allow those hydrogen ions to leak back into the stroma and drive that ATP synthase to make some ATP. And this, of course, should sound familiar because it's exactly the same kind of modus operandi of the good old mitochondrion. That's the structural aspect of photosynthesis in the chloroplast. We need to also talk about the metabolic or chemical reactions of photosynthesis itself. The pathway can be broken up really into two sets of reactions. There are what are called the light reactions. These are light dependent because they're going to take the actual energy from the light itself and convert that into usable chemical potential. So we're going to end up with molecules like ATP and NADPH. NADPH is one of those reduced molecules. It's going to kind of store electrons for later on. If you don't know what I'm talking about, go back and watch the notes casts on electron transport chain and the rest of respiration. The other part of photosynthesis is called the Calvin cycle. Now, these used to be called the dark reactions. We don't call them that anymore because it's not like they require the dark, like the light reactions require light. It's just that these are light independent. The Calvin cycle is kind of going on all the time. The Calvin cycle is involved with taking some of the energy that's produced in the light reactions and using that energy to fix the carbon dioxide into good old C6H12O6 or glucose. Taking a closer look at the light reactions, these are essentially an electron transport chain, just like we saw in aerobic cellular respiration. It's going to involve similar proteins that are embedded in the inner membrane of the thylakoids. It's going to involve an electron acceptor. In this case, it's NADP, which yields NADPH. That's going to go into the Calvin cycle. We'll talk about that here in a moment. But the overall result is the same. It's a proton gradient that's generated across that inner membrane, because in that inner membrane of the thylakoid is where we're going to find that ATP synthase. So as those protons leak back across the membrane, they're going to drive the production of ATP. Quick review of electron transport, just as a comparison's sake. This is taking place in the mitochondria in order to use the movement of the electrons to generate that proton gradient to make ATP. In this case, the electron carrier is going to be NADH, and we're going to ultimately generate water as the final electron acceptor. There is oxygen. Now let's compare that to the electron transport chain of photosynthesis, where instead of taking an electron from a food molecule and transporting that through intermediates, we're taking an electron that's been excited by light and transporting that through intermediates in order to create the proton gradient that's going to make ATP. In this case, we're using NADPH to transport the electrons, and instead of producing water, water is actually going to be split, and the hydrogen is going to be involved in the proton gradient, but it's going to yield oxygen instead. To do a quick comparison here of photosynthesis and respiration, the power supply is really the big difference, right? In photosynthesis, it's sunlight, where in respiration, it's the breakdown of glucose. But they're both using those power supplies to move electrons, to run some sort of a pump that's going to pump protons, build up a gradient across a membrane, and so that as that gradient is kind of relieved back through the membrane, it's going to drive ATP synthesis to stick that inorganic phosphate onto the ADP and to make ATP. A little bit more on the pigments of photosynthesis themselves. There are more than one. It's not just chlorophyll, and even so, there's more than one chlorophyll. There's chlorophyll A, B, and C, but there are other pigments, and they're always there. We just don't always see them. They're covered up by the kind of dominant chlorophyll. We could see them, especially up here in the Northeast during autumn, when as the chlorophylls kind of dry up and break down, the beta-carotenes and the fucoxanthins are much more visible, and that's why we get all the brilliant different colors. But those pigments are all embedded within the thylakoid membranes in kind of these groups of compounds, proteins, and the like, called photosystems. There are two different photosystems in most plants, photosystem 1 and 2, and those pigments get excited when light of a particular wavelength is shown on them. Just a quick review here of what I mean by wavelengths. Remember, light is part of the electromagnetic radiation spectrum. It's just the part that we can see, and if we break up the light into its individual wavelengths, we get to see all the colors of the rainbow, the reds, the orange, the yellow, the greens, the blues, the indigos, and the violets. Good old Roy G. Biv. But each individual photosynthetic pigment is excited by light at different wavelengths. It's not easy being green. Whenever we look at something, the color that we see is really a result of the light interacting with the surface of whatever it is that we're looking at. If that surface absorbs some wavelengths, then those are the wavelengths that we're not going to see. So what we're actually going to see is what is reflected, what is not absorbed. When we start to analyze the wavelengths of light that the different photosynthetic pigments absorb, we develop what are called absorption spectra. And it tells us which wavelengths they do absorb. Now, chlorophyll A, the primary photosynthetic pigment, is really good at absorbing the red and the blue ends of the visible light spectrum. It's pretty bad at absorbing there in the middle and the green, and so that's what's reflected, and that's why plants tend to look green to us. Now, there are other accessory pigments, like I said, that are there that are going to be better at absorbing some of the different wavelengths that chlorophyll A is not as good at. Those are the chlorophyll B and chlorophyll C and some of the carotenoids and xanthophylls and fucoxanthins and all of these different accessory pigments. Well, I think we'll stop there. That's probably quite enough. Again, thanks for checking in to Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. If you have any questions, feel free to drop me an email. Other than that, we'll see you in class.